So carrying on with the non-membranous organelles, so obviously ribosomes are not surrounded by membrane, they're just units of protein and rRNA. We come to the centrioles, and these are particular to animal cells, so we only find them in animal cells. And we find them, they're close to the nucleus. And they're always described as, they always occur in pairs, and they're kind of at right angles to each other, so they're kind of like that. So if these were cylinders, they'd be like that, always at right angles, at all sorts of angles, but always at right angles to each other. So these are described as paired cylinders. Now what you will see in particularly American videos is this term centrosome, and it describes the area in which they're found. So that includes the cytoplasm that surrounds them. The centrioles themselves are these paired cylinders, and you can see from the diagram that they appear to be made of long tubes, and that's not an accident. These long tubes in little groups of 3, 9 of them round the outside. So 9 groups of 3. There we've got sort of, we just colour it in and make it a bit more. So 1, 2, 3, 4, 5, 6, 7, 8, 9, and you've got 3 in each one. So they're not sort of a perfect, lovely circle. And they're made of long proteins called microtubules. So they're arranged, those microtubules, in groups of 3, 9 of them around the outside. If we look at them under an electron microscope, so remembering that we have to be able to recognise these down an electron microscope, obviously they've been sliced through. So what you're seeing is the end of this one. So as though you've sliced it through, I've not got anything really cylindrical, a lipstick. You're seeing that end with the groups of 3 microtubules. So here we can see 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3 microtubules. So we're looking at that end. If we slice down through it, then you're only going to see the microtubules that have actually been sliced through. And so we can see a bit of fuzziness. They represent the microtubules, say over here, and then the microtubules there and there that you can see. So it doesn't really look too much like a cylinder from there. So what do they do? Why do we have them? Well, it's a complete mystery as to how plant cells do this, because they don't have centrioles, but in animal cells they seem to be associated with making a spindle for the chromosomes to attach to during cell division. So what happens during cell division is that you get these little paired cylinders, I'm just going to draw them as T's, and they kind of elongate out to form a structure called a spindle, and that just gives somewhere for your chromosomes to attach to. And then those spindles are going to get shorter and drag the halves of chromosomes one end to the other. Again, we'll be doing cell division later on, so you'll find out all about it. Plant cells do it differently, but you don't need to know how.